Hello everyone. In this video, we're going to be covering a topic from Cambridge IGCSE Biology, the topic of movement into and out of cells and subtopic osmosis. Now, in this video, we're going to be covering these main topics. We're first going to be learning all about osmosis, what it is, how it works, and so on. And then we're going to be we're going to be investigating the effect of osmosis in plants. Then we'll be moving on to the importance of osmosis in plant tissues. And finally, the importance of osmosis in animal tissues. And these are the learning outcomes that we will be covering. So let's start on our subtopic for today, which is osmosis. Now, osmosis is another method of movement, another process through which substances move into and out of cells. And when we come to this topic of osmosis, you need to understand that osmosis is related only to the movement of water molecules. So it's a pretty simple process to understand. We can understand this well through this diagram over here. Now, in this diagram, what we see is that there is a cell, and we know that every cell is covered with a cell membrane. And this cell membrane of the cell happens to be a semi-permeable membrane. A semi-permeable membrane is also called a partially permeable membrane. So what osmosis is, like I mentioned, osmosis only focuses on the movement of water molecules. Now, as you can see in this diagram, we can see that inside of the cell, there are fewer water molecules. And outside of this cell, in this region outside, we can see there are more water molecules. Water molecules are given by these little blue circles given over here. So we can see that outside the cells there are more water molecules, and inside the cells there is less of these water molecules. Now, when we talk about the amount of water molecules, we talk about this concept of water potential. Now, a region that is having a lot of water molecules is said to have a higher water potential, while a region having less water molecules is said to have a lower water potential. And how osmosis works is that water molecules move from a region of higher water potential into the region of lower water potential, passing through this partially permeable membrane. So that's exactly what osmosis is. Let's take a look at the definition of osmosis given over here. So osmosis is the diffusion of water molecules through a partially permeable membrane from a region of high water potential to a region of lower water potential. So in simple terms, osmosis is simply the diffusion of water molecules from a region where there is more water, from a region of higher water potential to a region of lower water potential where there is less water, while passing through a partially permeable membrane. That is simply what osmosis is. Now, when we take a look at this osmosis definition, we come across a few new terms for y'all. For example, this might be the first time you're coming across this term partially permeable membrane and water potential. So let's start off with understanding first what partially permeable membrane is. Now, a partially permeable membrane is a membrane that allows small molecules like water to pass through, but not large dissolved solutes. So in this example, in this diagram, we see that this cell membrane happens to be a partially permeable membrane. And this cell membrane allows the passage of tiny molecules, such as these water molecules. So these water molecules are allowed to pass through this cell membrane and move into the cell. However, this partially permeable membrane would not allow larger molecules such as these solute molecules to pass through. So that is what we call a partially permeable membrane. It allows certain molecules to pass through, while it would not allow certain other larger molecules to pass through. And when we talk about water potential, which is also a part of this definition, water potential is a property of a solution that describes how free the water molecules are. So if there are a lot of solutes dissolved, the water potential is low. And if the solution is very dilute, the water potential is high. So like I mentioned, if you have a dilute solution with a lot of water molecules, that would mean that this solution has a higher water potential. Whereas if you have a solution with a highly concentrated solution, which is having a high concentration of solutes and less water, that would mean that that has a lower water potential. All right. And in the case of osmosis, it's always the case where the water molecules move from a region of higher water potential to a region of lower water potential while passing through a partially permeable membrane. That's what osmosis is about. All right. Okay, so let's move on to the next slide where we can understand better about osmosis through this diagram given over here. Now, what do we see in this diagram? Given here in this diagram, they've given us a partially permeable membrane. So there's a partially permeable membrane that I'm labeling over here, partially permeable membrane. And if you take a look at this partially permeable membrane, there are two different solutions on either side of it. On the left side of this partially permeable membrane is a solution with more sugar and less water. So less water would mean that it has a lower water potential. All right. So we have on this side, and I'm going to note that down, on the left-hand side is a region of lower water potential. So this region has a lower water potential. And on the other hand, if you take a look at the right side of this partially permeable membrane, you see a solution over here, which is said to have less sugar and more water. So more water would mean that it has a higher water potential. So I'm just going to mark that on this diagram. This region, this solution has a higher water potential. And now that we've learned what osmosis is, we know that when there is one solution having a higher water potential and another solution having a lower water potential that is separated through a partially permeable membrane, we know that osmosis is going to take place and these water molecules are going to move from this region with the higher water potential to the other region with the lower water potential. So that's exactly what happens here. Water molecules are going to move from the right side to the left side. And as a result, you'd see that the volume of the solution on the right side is reducing because of the movement of water molecules out of this solution. And when these water molecules move into the solution on the left side, you'd see the volume of this side increasing. So this is a simple setup that we can understand osmosis through. All right, osmosis is the movement of water molecules from the region of higher water potential into the region of lower water potential through a partially permeable membrane. All right. Okay, so now that we've learned the basics of osmosis, let's now move on to a practical where we're going to be investigating the effect of osmosis on plant tissues by immersing them in solutions of different concentrations. And this is a very famous practical. So you might have even come across this before. This is a practical where we take a potato. So we are taking a potato. I'm just going to make a simple drawing of this practical on this side over here. So we're taking a potato and we're cutting cylinders from this. So we're cutting like different slices. All right. And the first thing we do after cutting off these potato cylinders is we need to weigh them. We need to weigh them and find the initial weight of each potato cylinder. Each one is weighed separately. And what we do next is that we take these potato cylinders and we immerse them. Each one is immersed into a test tube containing a solution. So one test tube is containing distilled water, while the other test tubes contain sugar solutions of different concentrations. So what we're doing is we're taking each potato cylinder that we have weighed and we're immersing it into these different solutions, and we're keeping it this way for 20 to 30 minutes. So we're keeping them in these solutions for roughly about 30 minutes. And once the 30 minutes are complete, what we do is we remove these potato cylinders, we dry them to remove the excess liquid. And what we do next is we re-weigh them. We find the weight of them once again. So remember that we found the initial weight at the beginning, and then after immersing them in these solutions for 30 minutes, we're finding the weight of each potato cylinder again, the final weight of it. All right. And what we see is that our observation is that the potato cylinder that was immersed in distilled water will have increased its mass the most. So what we find out after doing this practical is that we see that some potato cylinders have increased their mass, while some potato cylinders have decreased their mass, and some potato cylinders have not changed their mass at all. And we noticed that the potato cylinder that was in distilled water has increased its mass the most. All right. And the potato cylinder in the strongest sucrose concentration will have decreased its mass the most. So the potato cylinder that you had put into the strongest sugar solution, the most concentrated sugar solution, has decreased its mass the most, while if there is a potato cylinder that has neither increased nor decreased in mass, it means that there was no overall movement of water into or out of the potato cells. All right. Okay, so you get a better understanding of this practical once we go on to the next slide. So this diagram over here is showing us exactly what we are doing in this practical. We're taking the potato cylinders and we're placing them in different solutions. One of it, like I mentioned, is distilled water, while the others are all different concentrations of sugar solutions. And like I mentioned, what we notice is that some of these potato cylinders have increased in mass, while some of them have not changed in mass. Their weight has not changed. And some of these potato cylinders have decreased in mass. So what's the explanation here? How is it that some of them have increased their mass, some of them have decreased their mass, and some of them have not changed their mass at all? So the explanation for this practical is osmosis. You see, if there has been a potato cylinder that has been placed in a solution with a higher water potential, if it was placed in a solution with higher water potential, that would mean that water has moved into that particular potato cylinder, which has resulted in an increased mass in this potato cylinder. But on the other hand, if this potato cylinder had been placed in a solution having a lower water potential, that would mean that water would have moved out of these potato cylinders and into the solution, because that's what osmosis is. Osmosis is where water moves from a region of higher water potential to a region of lower water potential. So if this solution happened to have a lower water potential than that of the potato cells, water is going to have moved out of these cells, and that would have resulted in a decreased mass. All right. And on the other hand, if there happened to be a potato cylinder that has not changed in mass, that means that there has been no net osmosis, no net movement of water into or out of these potato cylinders. All right. So that's the explanation. And this is a very famous experiment that helps us to get a better understanding of osmosis. All right. And now in our next slide, let's actually go into a more detailed explanation of how these cells behave when they are placed in different types of solutions. So when we're talking about osmosis and when we're talking about different types of solutions, we talk about three types of solutions, namely hypotonic solutions, isotonic solutions, and hypertonic solutions. Now, these might seem like pretty new terms to you all, so let me explain what each of these solutions mean. Now, a hypotonic solution is basically a very dilute solution. And we know that a very dilute solution happens to have a higher water potential. So I'm just noting this down over here. A hypotonic solution is a dilute solution that has a high water potential. And on the other hand, if we take a hypertonic solution, which is the opposite of hypotonic, a hypertonic solution is a very concentrated solution, a very concentrated solution. And we know that concentrated solutions usually have a lower water potential. A lower water potential. And then in the middle of these two is isotonic solutions. Isotonic solutions are solutions that have the same water potential as the cells. All right. So we're going to be taking a look at what happens to these potato cells or plant cells when they're placed in a hypotonic solution, an isotonic solution, and a hypertonic solution. So I'm just going to note the same thing down next to these diagrams as well. A hypotonic solution is a solution where there is a higher water potential in the solution compared to that of the water potential inside the cell. An isotonic solution is a solution having the same water potential, and a hypertonic solution is a solution having lower water potential. And now that we have learned the basics of osmosis, we know the simple rule of osmosis is that water molecules move from a region of higher water potential to a region of lower water potential. So in the case of a hypotonic solution, we'd see that the water molecules move from the region of higher water potential to the region of lower water potential. So basically, water molecules move from the outside to the inside of the cell. And when water molecules move into the cell gains a lot more water. And we say that when this water all comes into the cell, this water, this excess water, creates a turgor pressure. Now, a turgor pressure is the pressure created by these water molecules that push against the cell walls of these cells. So a cell that has got a lot of water in it is said to have a high turgor pressure. And this type of a cell that has a high turgor pressure is said to be turgid. So that's what happens when we place a cell in a hypotonic solution. The result is that this cell would become turgid. All right. And next, let's move on to the scenario where a cell is placed inside an isotonic solution. Now, like I mentioned, an isotonic solution is a solution where the solution has the same water potential as the inside of the cell. So in this case, when the outside and the inside is having the same water potential, there's going to be no net osmosis. There's not going to be a net osmosis, and there will be no net movement of water into the cell or out of the cell. All right. So what that means is that this type of a cell, which is placed in an isotonic solution, would not change at all. There would be no net movement of water molecule. So these cells are just going to stay as they are. All right. And then now we're coming to the final scenario of what happens when a cell is placed in a hypertonic solution. Now, like I mentioned, a hypertonic solution is a solution where there is a lower water potential compared to the inside of the cell. And we know the rules of osmosis. What happens is when there's a region of higher water potential and a region of lower water potential, that's going to mean that water is going to move from the inside to the outside. So basically what that means is that the cell is losing water. Water is moving from the inside of the cell to the outside of the cell because of the difference in the water potential. And as a result, this cell is going to become plasmolysed. Now, this term plasmolysis or plasmolyze means that the cell membrane tears away from the cell wall. As we can see in this diagram, the cell membrane has torn away from the cell wall and the cell has become shriveled and plasmolysed. So such type of a cell is what we call this type of a cell a flaccid cell. All right. So that's what you need to know. When a cell is placed in a hypertonic solution, it becomes plasmolysed and it becomes a flaccid cell. All right. And this is how plant cells behave when they are placed in these different types of solutions. All right. And now that we've understood this, let's move on to the importance of osmosis in plant tissues. Where does osmosis become important in the different parts of plants? So when we're talking about the importance of osmosis in plant tissues, now we've learned the basics about how plant cells behave with regard to osmosis. Now let's imagine a plant cell. Let's imagine a plant cell where there is a lot of water molecules inside the cell and this cell has become turgid. So what I'm drawing here is a turgid cell, a turgid plant cell. Now, when we have a turgid plant cell such as this means that this cell has a high turgor pressure. And like I mentioned, turgor pressure is the pressure of the cytoplasm pushing against the cell wall. Now, when there is a turgid plant cell with a high turgor pressure, this pressure prevents any more water entering the cell by osmosis, even if it is in a solution that has a higher water potential. So once plant cell has become turgid, even if this plant cell is placed in a solution with a higher water potential, this turgor pressure would prevent any further water, any more water from entering into the plant cell. So this basically prevents the plant cells from taking in too much water and bursting. Now, this process becomes particularly important in the case of root hair cells. Now, we've learned about root hair cells in a previous video. The root hair cells are the cells in the roots of plants that give rise to the root hairs. And we know that these root hair cells are surrounded by soil water. So there's a lot of water around these root hair cells. I'm drawing water in blue. So there's a high water potential, a lot of water around these root hair cells. All right. So I'm just going to label that as well. Outside the root hair cells, there is a region of higher water potential. And comparatively, inside the root hair cells is a region of lower water potential. Now we know what happens then when there are two regions with different water potentials. Water is going to move from the region of higher water potential into the region of lower water potential. So water is going to move from the soil water into these root hair cells by osmosis. So the water moves across the cell membrane of root hair cells into the root by osmosis, and then water moves across the root from cell to cell by osmosis until it reaches the xylem. So this water that has entered into the root hair cell moves from cell to cell until it reaches the root xylem. And once the water has entered the xylem, it is transported away from the root by the transpiration stream, and it goes across the xylem vessels, right? So this is an instance where osmosis is important in plant tissues. Osmosis is the process that allows the water molecules in the soil water to move across the partially permeable membrane of these root hair cells and enter into these cells. And this is how the plant gets its water, through the process of osmosis, where water molecules move from the soil water into the root hair cells. All right. So now that we are done with that, let's also look at scenarios where osmosis becomes important in animal tissues and animal cells. All right. Now, when we talk about animal cells, we know that animal cells, unlike plant cells, they do not have cell walls. All right. So animal cells also lose and gain water as a result of osmosis. However, as animal cells do not have a supporting cell wall, the results on the cell are more severe. All right. So now I'm going to explain to you what happens when we take animal cells and we place them in a hypertonic solution, an isotonic solution, and a hypotonic solution. And the example of the animal cell I'm taking are red blood cells. All right. Now, I hope you remember these terms: hypertonic, isotonic, and hypotonic. A hypertonic solution is a solution which has a lower water potential. So I'm just noting that down over here. An isotonic solution is a solution having the same water potential as the cells. And a hypotonic solution is a solution having a higher water potential compared to the cells. A higher water potential. So let's see how these red blood cells would behave when they are placed in these different types of solutions. So let's start off with the hypertonic solution. When these red blood cells are placed in this solution where there is a lower water potential, we know what's going to happen. When the outside is having lower water potential, that means that water is going to move from the inside and come to the outside of the cell. Or in other terms, these cells are going to lose their water because the water is going to move from a region of higher water potential to a region of lower water potential. So what happens to these red blood cells when they lose water in a hypertonic solution? What happens is that they become cremated and shriveled up. So you see this diagram over here, you see these red blood cells that have lost water, and as a result, they have shriveled up and become crenated. So that is what would happen if you place an animal cell in a hypertonic solution. All right. Now moving on to how it works in an isotonic solution. When we have an animal cell in an isotonic solution, since the inside and the outside are both having the same water potential, we know that there's going to be no net osmosis, and as a result, there'll be no change in these red blood cells. All right. So in an isotonic solution, there would be no change in these cells, right? Then finally, let's see how these red blood cells would behave in a hypotonic solution. In a hypotonic solution, what would happen is that water would move from the outside, from the higher water potential, to the inside of the cell. And as a result, when these cells gain a lot of water, the cell membranes of these cells become stretched too far, and you'd see that these cells end up bursting. So when these cells are placed in a hypotonic solution, water moves into these cells and the cell membranes become too stretched out, and that would finally result in these red blood cells bursting. So this is how the animal cells behave in the three different types of solutions. And with this, we have come to the end of osmosis, and we have covered everything we need to know about osmosis. So now that this is done, let's try out a few practice questions and to see whether we've got the correct understanding of osmosis. All right, so here's our first question. The diagram shows water and sugar molecules on either side of a partially permeable membrane. So what they have shown here is that there's a partially permeable membrane. On one side is region Y, and on the other side is region X. Now, region Y is a region having a lot of water molecules, as you can see, a very dilute solution. So a dilute solution is said to have a higher water potential. And if you look at region X, you can see that this region is having lesser water molecules. So it's a more concentrated solution. So we say that it has a lower water potential. Less water molecules means a lower water potential. So now that we've learned all about osmosis, we know how it works. Osmosis is where water molecules would move from the region of higher water potential to the region of lower water potential across the cell membrane or a partially permeable membrane. So that's exactly what's going to happen. The water molecules would move from region Y to region X. There's going to be a net movement of water molecules from region Y to region X across the partially permeable membrane. So if we read through these answers, we'd see that this answer is given in answer D. More water molecules pass through the membrane from Y to X than from X to Y. That's the net movement from Y to X. All right. Okay, so now let's try out one more question, one more practice question. Here it is. On a dry, sunny day, how does water vapor move through the stomata of a leaf? So we know the basic structure of a leaf. If you take a leaf, on the underside of the leaf, there are these stomata pores through which gases can move about. So on a dry, sunny day, that would mean that the atmospheric air has a lower water potential, while the inside of the plant leaf has a higher water potential. So what that would mean is that water would probably move out from the region of higher water potential to the region of lower water potential. So water is going to move from the inside of the leaf to the outside of the leaf through osmosis. So the answer given out of A, B, C, D, the answer would be that water moves out of the leaf by osmosis. All right. And now that we're done with the practice questions, let's also try out one more question from the question bank before we wrap up. All right, so here's a question that we can try out. The question says that the figure illustrates two cells where the net movement of water in the direction indicated is in the direction indicated by the arrow. So there are two cells, cell X and cell Y, and the diagram is showing that water is moving from cell X into cell Y. And the question is, what causes water to pass from cell X to cell Y? So the answer would obviously be that cell X has had a higher water potential and cell Y has had a lower water potential. So I'm going to write that down. The fact that water is moving from cell X to cell Y means that cell X has had a higher water potential, while cell Y has had a lower water potential. And for that reason, because of that, water has moved from cell X to cell Y through osmosis. So that's the answer I need to choose. And the answer is answer 2. Water potential is higher in cell X and osmosis occurs. So this brings us to the end of this video. That's all we need to know about this subtopic on osmosis. I hope all of you got a good understanding on this.